Thanks for this opportunity to be here with you. I will introduce myself. First, I am Sally Farouk from National Research Center from Egypt. Before starting, I want to tell you that this presentation is about two different studies that were merged together according to the recommendation from the organizing committee. So it's supposed to introduce this presentation with Professor Noha Badreddin, but she had an emergency issue preventing her from joining us. So I will present our talk now. Okay. First, the title was modified for the merged talk. So it will be evaluation of microRNA 374 and tumor suppressor microRNA 7 and 15, expression in Egyptian colorectal cancer patients, presented by both me, Sally Farouk, and Dr. Noha Badreddin from National Research Center, Cairo, Egypt. First of all, I will introduce microRNA as a small, or they are a small non-coding RNAs molecules of 18 to 22 nucleotides that act as regulators of gene expression by interacting mainly with messenger RNA. Currently, over 3,000 microRNAs are found in almost all human body fluids. These microRNAs are implicated in various processes and they play a significant role in progression and regression of several number of diseases like cancer. They exert effects on the messenger RNA by inhibiting translation and degradation of target messenger RNA. They can act as post-tumor suppressor and onco mRNAs. The role of microRNAs in cancer, they elicit either oncogenic or tumor suppressor function according to the via silencing their target genes. So in case of cancer, if the microRNA upregulated, oncomicroRNA, if upregulated, this will lead to tumor suppressor messenger RNA inhibition. So this will inhibit tumor suppressor genes, and this will lead to carcinogenesis and cancer progression. In the same case, if tumor suppressor microRNA downregulated, this will lead to oncogenic messenger RNA stimulation and overexpression, and this will lead to oncogenes stimulation and activation, and this will lead also to carcinogenesis and cancer progression. So here I'll talk about microRNAs and colon cancer. So cancer can be carcinogenesis can be occurs via several types of pathways such as tumor promoting inflammation, genome instability and mutation, immortal replication, apoptosis, metastasis, angiogenesis, growth suppressor evasion, and proliferative signaling pathway, and so on. Now I will talk about colorectal cancer epidemiology. Colorectal cancer is the third most common cancer worldwide, accounting for approximately 10% of all cancer cases, and is the second leading cause of cancer-related death worldwide. It's predominantly affect older individuals, with the majority of cases occurring in the people from age 50 and above. Several lifestyle factors contribute to the development of colorectal cancer, such as high intake of processed red meat and low intake of fruit and vegetables. Sedentary lifestyle, obesity, smoking, and excessive alcohol consumption will lead to progression and incidence of colorectal cancer patient or CRC. Large geographical variations in incidence and mortality rates were observed. The incidence rate were highest in Europe and Australia and New Zealand, and mortality rates were highest in the Eastern Europe. Now I will talk about role of our microRNAs. First, microRNAs 374 in colorectal cancer. They have three rules: regulation of cell cycle and regulation of epithelial mesenchymal transition and interaction with TGF-beta. The role of microRNA 7 in colorectal cancer Mainly, it is a tumor suppressor, has a tumor suppressor role, and also has a regulation of epithelial mesenchymal transition via targeting twist1 pathway and the PAC. Also, it can be a tumor suppressor via expression of several oncogenes, including EGFR, epidermal growth factor receptors, and RAF1. The last microRNA here, we are talking about microRNA 15A in colorectal cancer. They had a tumor suppressor role as microRNA 7 and had a metastasis role in metastasis regulation role and help in angiogenesis regulation. Aim of work, I will talk about from this point, I will talk about the two studies differently. Our first study mainly aimed to explore the differential expression levels of microRNA 374 in sera of Egyptian colorectal cancer patients. Moreover, to explore its correlation with TGF-beta 1 and determine the diagnostic efficacy of their circulating expression levels. For the second study, we investigate the role of tumor suppressor microRNA 7 and 15A, investigate the relative expression in Egyptian colorectal cancer patients, and assess their clinical value as a prognostic and diagnostic biomarkers. Methodology here for study for microRNA screening for microRNA 374 relative expression. This study conducted by 150 subjects. We had a healthy control group of 50 participants and colorectal cancer patients of 100 patients. Serum samples was withdrawn from post-control and CRC. Then microRNA extraction were done, followed by RNA concentration measurement by using nanodrop. Then cDNA reaction was done, followed by Q real-time PCR. This circulating and also circulating TGF-beta serum level were measured by ELISA. Our results here regarding the circulating microRNA 374 and TGF-beta serum level. This figure show a highly significant upregulation of circulatory microRNA 374 in serum of CRC patients compared with control group. Also, there is a higher significant levels of circulating TGF-beta 1 in serum of CRC patients compared to control group, with different p-values as shown in significantly p-values as shown in this table. Row curve analysis was done to measure diagnostic efficacy of circulating 374 microRNA 374. Here, this figure shows that circulating 300 microRNA 374 has a significant diagnostic efficacy. To differentiate between CRC patients and healthy subjects with area under curve equal 0.94, with high specificity that about 97.14% and sensitivity 83.72%. Now, regarding the other two microRNAs, microRNA 7 and microRNA 15A relative differential expressions, the methodology here was different than the other. We study the expression of microRNA 7 and 15A in tissues and in serum of CRC patients and controls. This study conducted by 100 subjects. 50 subjects as control group and 50 as CRC patients. The tumor tissues and adjacent cancerous tissues samples were taken from CRC patients who underwent colonoscopy. Also, serum samples withdrawn from both control and CRC patients. For tissue sample, it was first homogenized by tissuealyzer before microRNA extraction. Then from this step, serum samples and tissue after tissue lasers go through microRNA extraction. That will followed by RNA concentration measurements by nanodrops. Then cDNA reaction was done, followed by urea time PCR. Then data analysis for the serum. So our results here regarding post microRNA 7 and 15A, our results showed that post microRNA 7 and 15A were significantly downregulated in tumor tissues compared with adjacent non-cancerous tissues in microRNA 7 and microRNA 15 with higher significance p-values 0.02, 0.02, and for the also in serum samples, microRNA 7 and 15A were significantly downregulated in CRC patients compared with a control group. Then root curve analysis was done to measure diagnostic efficacy of both circulating microRNA 7 and microRNA 15. This post figures shows that circulating microRNAs 7 and 15 had AUC area under curve value 0.89 and 0.75 with specificity 85% and sensitivity 70.59% for microRNA 7 and the specificity 83.33% and 60% sensitivity for circulating microRNA 15A. So they had ability to differentiate between CRC patients and healthy subjects by these results. Conclusion, circulating microRNA 374 and the TGF-beta can be potential non-invasive, can be used as a potential non-invasive diagnostic biomarker for colorectal cancer patients, as well as microRNA 7 and microRNA 15A. For further studies, it can be a therapeutic target for colorectal cancer patients by stopping or make antisense for these microRNAs to be a therapeutic target for colorectal cancer patients further on. Thank you all for your listening now, and any questions if you want.
Thank you, Dr. Farouk. That was wonderful. We do actually have some questions in the chat for you about the role of these miRNAs a little bit further. So one of our first questions is, why do you think these miRNAs are actually like exiting your tissue biopsies and circulating? Do they have some sort of functional role in circulation?
Yes, they have a functional role in circulation via targeting specific genes by silencing their messenger RNA. In our study, we prefer to do microRNAs in tissues and confirm our results by seeing them in serum, because in previous studies we had shown some microRNAs were expressed in serum and not expressed in tissues, and we want to know if this tumor suppressor microRNA if they are expressed both in tumor and tissues and serum, or not serum only or tissues only, according to our population.
I see. And another question that comes up when you talk about microRNAs, these are broadly known to be tumor suppressive in a number of cancers. So we're wondering, with your expertise on mRNA 7, 15A, and 347, have you seen these pop up in any other cancers? And do you have any ideas what they might be doing there?
Yes, not each cancer type have had several specific genes and specific pathways. In colorectal cancers, microRNA 7 and 15 act as tumor suppressor. They may act this function in other cancers, but it may be upregulated and be oncogenic microRNAs in some type of cancers. It will be different. Also, in the same type of cancer, population differs. The type of populations, the race will be affect microRNA expressions, and several studies talk about this point of view and revealed that microRNA expression varies from one population to another and from race to another, even in the same type of cancer.
That's very interesting. Have you thought about outside of Egypt what other populations this might be a good biomarker in based on literature?
In Egypt, we study these two microRNAs or three that there is no publications not yet published regarding these microRNAs, but 374 act as tumor suppressors in other populations, and here in Egypt, it acts as oncomicroRNA and highly expressed. This is unpublished data, and the complete finishing our work, we will publish it, and this will reveal that huge and different results we had here, that microRNA 374 was highly expressed in our population rather than European population or American population. Several previous studies revealed that it was a tumor suppressor microRNA.
I see. Very interesting. So sort of the flip side of the same question that I'm asking is, are there other tumor suppressor mRNAs within CRC that, maybe we didn't talk about today, but you also think might be implicated or important to study?
Yes, of course. There are several types of cancers that act as a tumor suppressor, like microRNA 27. I worked before and published a publication about it. Also, microRNA 15B, microRNAs 34B. They are they are a lot of them may act as tumor suppressor genes.
Got it. Thank you. So another question that we have coming up is, do you think that you can actually identify the critical cells that are being targeted by these microRNAs?
Yes, we can. Your questions about confirmation our targets genes?
We're
Please
trying to
rephrase the
understand.
question.
Yeah, of course. So we're trying to understand that, when you have these miRNAs that are in the circulation, they're probably going to be affecting certain cells more than others. Are you aware of what those critical target cells might be?
This will our next approach in lab for studying them in the cell lines, and try to silencing or knock out these microRNAs to prove or to focus on specific pathway to stop it. And this is as a target therapeutic target for our research.
Got it. Yeah, that's wonderful. And one slightly different themed question, but very interesting that's come up is that a lot of these microRNAs that you're talking about are actually located on the X chromosome. So are you guys detecting any sort of sex differential effects of the microRNAs?
They expressed in serum, not exosomes. They are free circulating microRNAs that I studied on, not exosomes. In
Right.
tissues, in free circulating microRNAs.
Got it. But a lot of these microRNAs, are they on their actual genetic background, they're located on the X chromosomes, is that correct?
Yes, yes,
So
yes.
so in that case, given that you have the XY difference, are you seeing any differential effects based on women and men patients that you see?
Women and man patients. I am not studied this. I am not focused on this point, but it may be differs. Maybe it's our next research to ask to answer your questions, but I have no idea.
Sure, of course. Well, thanks for taking these questions. I think I want to end with just one more question that we have from Mustafa here. And
Okay.
so he's asking, a previous study showed that microRNA 378 targets succinate dehydrogenase complex. Can you talk about that a little bit?
About please, I can't understand the question.
Sure. So in terms of microRNA 378, which is a different microRNA in your case, it's been seen that that microRNA targets succinate dehydrogenase complexes. So have you seen any sort of similar effects in microRNA 374?
No, 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 no. No, I will focus about 374 and its effect on pathway of TGF beta pathway and not focusing on other pathways.
Got it. Okay.
So
Well,
I didn't can't answer this question.
yeah, well, it's always nice to get all these range of questions, and I encourage you to always also take a look at the chat for any remaining questions to be answered.
Okay,
But thank you so much for your talk.
thank you. Thank you very much.